Hello dear students, welcome to Biochemistry Byrevati. I am Revati and in today's class we will learn an interesting and a very important topic, that is the concept of stereochemistry. Stereochemistry, it is a 3D language of chemistry and it explains why the two molecules have the same formula, uh, can behave very differently from each other, means one might heal you, the other might harm you. So by the end of this video, you'll come to know that what is chirality, enantiomers, diastereomers, and why stereochemistry is essential in medicine and biology. So let's get started. Stereochemistry. It is the study of three-dimensional arrangement of atoms in a molecule. So what is stereochemistry? It is the study of 3D arrangement of atoms in a molecule. Now why is this important? Means the arrangement of atoms in a molecule, that too in a 3D form, because in our body molecules don't exist in a flat form, right? They do exist in a which form? They do exist in a 3D space. So here the molecules in our body, they don't exist as a flat. If you take it on a plane of paper, the molecules don't exist flat on a plane of paper, but they do exist in a 3D space, right? If you take two molecules having for example, if you take two molecules having same molecular formula, same and even the same bonds, but they still act differently in our body. Why they still act differently? Why they still act differently in our body? Because the arrangement of atoms, are different in a space. Is it clear? To understand stereoisomers, first we have to know, first we need to know what isomers are, right? So if you take types of isomers, they have been categorized into two. One is structural isomers and stereoisomers. Structural isomers, they have same molecular formula but different connectivity. Let's see how. By taking example. So example, ethanol or ethyl alcohol and dimethyl ether. So both these compounds have molecular formula has C2H6O, but their connectivity is different. Let's see in the structure form. First, ethanol or ethyl alcohol. So if you take these two carbon atoms, 1, 2, 3, 4, 5, 6 hydrogen atoms, C2H6 and O, right? So here if you see, you will have the functional group as hydroxyl. So it has hydroxyl group as a functional group. If you take another example, dimethyl ether, it is C1, C2 carbon. Again, if you see hydrogen atoms 1, 2, 3, 4, 5, 6, and this one oxygen. So here you will have a functional group as ether. So This is the different connection, means there is a different connection of bonds between what? Ethanol or ethyl alcohol and dimethyl ether. So this is an example for what? Structural isomers, right? Isomers, if you see, they also have same molecular formula, same connectivity, but there is a difference in the spatial arrangement of atoms, right? Again, in this you have, it is categorized into enantiomers and diastereomers. About this we'll discuss in further. Now we come to the concept of chirality. What is actually chirality? A molecule can be chiral when it cannot be superimposed on its mirror image. Superimposed means placed. When it cannot be superimposed on its mirror image, right? Then we call it as chiral. So here, if you take example of right hand and left hand, right? Well, right hand can fit onto the left hand, not exactly, right? Or example, if you take right hand glove can fit onto left hand glove, left hand, will it? Not, right? So here that is nothing but the chirality concept. So the most common cause of chirality in organic molecules especially is what? Achiral carbon. Now what is this? Chiral carbon. Now what is this chiral carbon? When it is attached to 4 different groups, right? So if you take example as lactic acid, which has been connected to 4 different groups. So here now this carbon becomes chiral carbon. If you take example of achiral, it is acetic acid. Now what is achiral? When it has plane of symmetry. Is it clear? Chirality leads to stereochemistry, stereoisomers, right? So here chirality leads to stereoisomers. We have already studied about this, right? What are stereoisomers? They have same molecular formula, same connectivity, but different 3D arrangement, right? So again in this stereoisomers we have enantiomers and diastereomers. Now what are enantiomers? They are non-superimposable mirror images. And what are diastereomers? They are not mirror images. And here they have same physical property except for what? How they radiate light and how they interact with other chiral molecules. And diastereomers, they have different physical and chemical properties. Here if you take example, D-alanine and L-alanine. What is alanine? It is an amino acid. Here, glucose and galactose. If you take the structure, if the amino group is on left side, it is L-alanine. If the amino group is on right side, what it is? D-alanine. Got it? And for glucose, it is Fischer projection. About this we'll learn in further classes. Glucose, and this is galactose. So if you see the fourth configuration is different except the same. So these are the examples for diastereomers. Let's come to the topic optical activity. So here chiral molecules can rotate plane polarized light. Property called what? Optical activity. So here chiral molecule can rotate what? Plane polarized light. If the molecule rotates light towards clockwise, then it is dextrorotatory. If the molecule rotates light counterclockwise, then it is levorotatory. So here it is minus sign. Is it clear? So here this plus and minus signs represents behavior of the light. Means when the molecule interacts with light, how it behaves, right? This plus and minus represents. And here the D and L represents structure of the molecule. Is it clear? Next it is naming systems. Here we use this naming systems to describe what? Stereochemistry. So first is R-bias system. So according to Can, Ingold, Prelow rules and CIP rules in short. According to these rules, first we have to label chiral centers. Means carbon attached to different groups. rank the chiral centers by giving priority based on what? Atomic number. So here first number will be having the heaviest atom, right? Like that we have to give ranking. Third, give the ranking by 1, 2, 3, like order, right? So here lowest priority will be always given to hydrogen only because its atomic number is 1, right? So here like this, if it rotates, the order rotates, example, if the order rotates like this, means towards right, in Latin rectus means right, means if it rotates towards right, then it is R configuration. Is it clear? And if it rotates left or counterclockwise, so here it is clockwise, so here it is anti-clockwise or counterclockwise. In Latin, S stands for sinister. So this is S configuration. Take example of lactic acid. So first OH is given first, second carboxyl, third methyl, and finally hydrogen 4. If the order is 1, 2, 3, right? So now this is towards left, right? So it is S lactic acid. You see for R configuration, hydroxyl 1, carboxyl 2, 3. You see it is going towards right. It is R lactic acid. Is it clear? For DL system, it is mainly used for sugars and amino acids, mostly based on the reference molecule glyceraldehyde. If the important group OH or amino group is on right, then it is D molecule. If it is on the left, then it is L molecule. Let's take example of glyceraldehyde. If the OH group is on the right side, then it is D-glyceraldehyde. If the OH group is on the left side, then it is L Glyceraldehyde. Is it clear? Now comes to the biological importance. Why do we care so much about stereochemistry? Because life is stereospecific. You know why? Because proteins are made up of L amino acids, and the sugars which are produced in our body are mostly forms are D sugars, and the enzymes are like lock and key, means they connect to correct stereoisomer only. Is it? So like this, they are all important. Means the stereochemistry is very very important in our life. So if you take example of real life, thalidomide tragedy. Have you heard of this name? Thalidomide means this tragedy happened in the year 1950s or 60s. I don't remember much, but in this tragedy, one form of the drug had helped with morning sickness, and the other mirror image caused severe birth defects. Now, this disaster taught the scientists to how stereochemistry is crucial in designing the medicines. Means how much care should the doctors or scientists take in designing the medicines by going through this stereochemistry. That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the video, please comment on your doubts, right? And if you found this video helpful, please like, share, and subscribe to Biochemistry Byrevati for more simple and clear explanations. Please do support me. In the next video, we will continue with the same concept of stereochemistry only with reference to carbohydrates, right? So until then, keep learning and stay curious. See you in the next class.